The topic of my presentation will be the gut-immune connection. It's a topic that I recently covered extensively in my new book with the same title. So let me start out with the conceptual model that I have been using for some time now to explain the interactions, the complex interactions between the brain, the gut microbiome, and the immune system on the one side, and the environment on the other side. I'd like to continue this concept of the this systems view, that we can understand many of the problems that arise health-wise come from two major perturbation of the balance within the system. One are psychosocial stressors that act primarily on the brain, and dietary stressors that act primarily on the gut. So there's been a dramatic change in the view of the gut microbiome during the last decade. On the left side, you see the something that we have known for some time, namely of these many metabolic functions, capabilities of gut microbes, for example, breaking down complex fiber molecules, including resistant starch, into short-chain fatty acids. But there's many other things that microbes can metabolize, break down from unabsorbable large molecules that we get with our diet into smaller molecules which then can be absorbed, but also can have an effect on our body. If you go to the right side, this is the more recent insights that we've gained, that many of these metabolites and components of the gut microbiome can actually affect pretty much all the organs in our body and organ systems, including the nervous system of the gut, the cardiovascular system, the brain, the immune system, topic of this presentation. But also, what I come to the end of my presentation has some effect on the pulmonary system. We know that there is a communication between gut microbes and some of their metabolites, like again, the short-chain fatty acids, or metabolites of amino acids that we ingest with our diet, modulating the immune system and, for example, playing a role in the susceptibility and also the resilience to inflammatory conditions of the gut, such as inflammatory bowel disease. Another important implication of the interactions of gut microbial metabolites and cell wall components is with the immune system of the gut in terms of playing a role in the recent rise of food allergies and food sensitivities. Very important cardiovascular disease, certain metabolites of the gut microbes, like this a substance called TMAO, has been identified as a major risk factor for cardiovascular disease. And then finally, the important connections with the brain. Again, the main players being the one side immune mediators, on the other side short-chain fatty acids, which have as in general, an anti-inflammatory effect. So it's just a snapshot of the complex interactions that have been unraveled between the gut microbiome, the immune system, and many or most of our organ systems. The gut microbes are quite an intelligent part of planetary life. They have up to 20 million microbial genes, outnumbering by far our 20,000 human genes. And there have been interesting speculations that, have we been neglecting 99% of all this genetic information in a system where we live in close symbiosis with the gut microbiome, meaning that we're really one organism, and this organism is regulated by genes. The human genes, just being like this red line, is represented by this red line on the surface of this human figure, whereas the the microbial gene is much more expansive and has a lot more function, many of which we don't really know. So what are the signals that microbes can generate with this vast genetic storage of information that they have? Well, there's basically three types of these signals. Some are diet-derived, some are host-derived from molecules that our body produces, and some come from the microbial, from the microbes themselves, the microbial membrane, which has molecules in it that interact with our immune system. And let me say a few things about both the diet-derived and these microbial membrane-derived molecules. So in terms of the diet, this is illustrated here on we consider a healthy microbiome-friendly diet, which is predominantly plant-based. These nutrients, tens of thousands of molecules and phytochemicals, are metabolized by this vast number of microbes we have in our gut. You can see this generating a vast number of metabolites and signaling molecules, which then interact with our body and the different systems in our body, including the immune system and the brain. And let me just illustrate one of these examples. Just step back here again. One of these examples of a diet-derived signaling system that the microbes play a key role in. So this has to do with the essential amino acid tryptophan that we have to eat with our diet because our bodies cannot produce that molecule. And we know that, we know today, in just the last few years, that tryptophan can be broken down into serotonin, represented here with this abbreviation 5-HT, 5-hydroxytryptamine, and it's stored in cells in the gut. 90% of all the serotonin in our body is stored in these cells in the gut, and these cells communicate closely with the brain through the vagus nerve. But what's interesting is that this synthesis of serotonin is to a great degree influenced by signals from the microbes. And again, the short-chain fatty acids or secondary bile acids that stimulate a hormone in these cells to metabolize tryptophan into serotonin. There's another, so if serotonin is the good guy in this tryptophan metabolism, this microbial enhanced or mediated tryptophan metabolism, another group of molecules, the kynurenines, are the bad guys. So those kynurenine molecules can get into our central nervous system and into the brain and cause neuroinflammation and neurodegeneration. And then there's a third group of molecules that microbes play the key role in metabolizing, because our body does not have the molecules, the enzymes to break that for this particular pathway. And these are the indoles. The indoles, it's a group of molecules that can have both beneficial and detrimental effects on the brain. One particular member of this family, indoxyl sulfate, has been identified as an important player in several in autism spectrum disease and in Alzheimer's disease. So this is just illustrating the complexity with one molecule, tryptophan, our diet, that the microbes play crucial roles in generating signal molecules that affect the rest of our body. But ultimately, this interaction between the microbes and the gut-associated immune system has major effects on the immunity, not just in the gut, so defenses against infections, inflammation, but also throughout our body. And this is illustrated here, a figure from an excellent recent review that shows that immune signaling plays an important role in establishing this gut-liver connection. Let me just illustrate this one figure for the brain. So these cell wall components of certain microbes with this abbreviation, these molecular patterns that are cell wall components, such as lipopolysaccharide or flagellin or others, which activate receptors, so-called toll-like receptors on certain immune cells in the gut, particularly initially the dendritic cells, which then start producing cytokines. These cytokines can spread throughout the body, affect all the other organs, as I just said, but they also can cross the blood-brain barrier, activate immune cells in the brain, the microglial cells, which then amplify this inflammatory cascade. And these immune modulators, particularly cytokines, can then affect the activity, structure, and function of nerve cells, leading to neuroinflammation and neurodegeneration. So something that 10, 15 years ago, we didn't even conceptualize at all. There's something that originates in the gut, particularly with beneficial microbes in our gut, that if they get too close to immune cells, that it triggers a cascade that ultimately could play a role in some devastating diseases such as Parkinson's disease and Alzheimer's disease. And we know a lot of details from animal models, which cells in the brain, so from nerve cells to these various glial cells or endothelial cells that make up the blood-brain barrier, how they can be affected in a negative way by these cell wall components of microbes in the gut, but also in a positive way by these short-chain fatty acids. So these small molecules that are generated when microbes break down complex carbohydrates that they get in terms of dietary fiber in our diet. On a healthy diet, we have a large variety and diversity of the gut microbes. They are separated from the gut-associated immune system by two mechanisms. One, a mucus layer that does not allow these microbes to get close to these dendritic cells with their sensors, and then with a very tight epithelial barrier where these cells that make up the gut lining will not allow for any components of the gut microbes to get through and come in contact with the gut-associated immune system. So what happens in the leaky gut situation? First of all, a leaky gut can be the consequence, or is the consequence, of a chronic dietary and psychological stress situation. Both of these stressors or perturbations of the homeostasis can affect the system in a similar way. Give you one example, what we think has happened in the last 75 years or so after World War II with the accelerating industrialization, westernization in the world, changes in lifestyle, the early colonization of our gut with microbes related to antibiotic exposure, a lot of exaggerated hygiene measures, and then medical practices, particularly again, antibiotics. All these have led to a change in this gut microbial ecosystem. The changes are in decreased richness, abundance, and diversity. So the ecosystem has suffered in its resilience and resistance. But this altered gut microbiome, which has adapted pretty rapidly to these changes in 75 years, now interacts with our ancient gut immune system that is largely genetically determined. The end result of this is that it changes the gut-associated immune system with consequences throughout the body. So what has happened, again, over the last 75 years, and something I elaborate on in my books. So these chronic stressors, dietary and psychological stress, can be referred to as an increased allostatic load. Allostatic load means when a chronic stress constantly challenges these stress response systems, the neural system and the immune system. And instead of providing an adaptive response that protects us from these stressors, the stress response itself will become maladaptive, as shown in this chronic engagement of the immune system in an inappropriate way. So this, we can say, puts gut health and the gut and its interactions with the diet and its the immune system responses, puts this at center stage for this chronic disease epidemic, again shown here in red, the links that we can attribute to abnormal immune system activation. And I've added one more to this, which is not a non-infectious disease, but it's this vulnerability to viral disease that has become apparent during the pandemic and during the research that has been done on vulnerability to COVID-19 and particularly abnormal and more severe courses, disease courses after infection. And just as a reminder, COVID-19 affects primarily the lungs. It's not a disease that goes through the gastrointestinal tract primarily. So this is something where the gut affects a distant organ. And the summary of this paper showed that gut microbial composition in patients correlates with disease severity and plasma levels of systemic immune markers. The same markers that I said earlier are part of the systemic immune activation in all these, in this chronic disease epidemic. But to illustrate it in a visual form, so the effects, the changes in the gut microbiome that most likely were present in these individuals before they got infected in response to an unhealthy diet and chronic stress. Again, we have quite a bit of evidence that the patients particularly vulnerable to these more severe infections are under greater psychological and dietary stress, and that these changes in the gut then can affect the immune response in the lungs, and that may also play a role in some of these unexplained brain alteration or brain pathologies that have been described in some patients with COVID-19. Also, this entity of long COVID, this persistence of fatigue, brain fog, and cognitive dysfunction that so many people that had the infection, even if the infection was mild, complain about. I would like to speculate that they also are related to the effect of the exaggerated immune system activation in response from the gut microbial immune interaction, play a role in this as well. So there's lots of research that will have to be done still to corroborate some of these concepts, but I think it's pretty clear that the gut microbiome and its interaction with the gut immune system play a crucial role, not just in this chronic non-infectious disease epidemic, but also in the vulnerability to complications from COVID-19. So I would like to end this presentation with emphasizing the importance of human nutrition. We don't have medications at this stage or interventions that are able to target specific changes in these altered interactions in the gut. But we do know several important aspects of the story which have consequences of what we eat and how we look at our diet and our nutrition. Understanding how our diet and nutrition status influence the composition and dynamic operations of our gut microbial communities and the arms of our immune system represents an area of scientific need, opportunity, and challenge. It's my prediction that this will be a major focus of research in the next 20, 30 years. And these insights and the solutions that come from this will help to address and hopefully come up with solutions for a number of urgent global health problems. And let me end with a few take-home points from my talk First, communication between the gut microbiome and the gut-based immune system is an ancient, evolutionary conserved system that modulates the function of every organ, including brain structure and function. So the system itself is ancient, goes back in evolution to hundreds of millions, probably billions of years. What has gone wrong is what happened in our environment and how this system has reacted to this. This ancient communication system connects the mind, the gut, connectome, food, and the environment in a way that can not only best, but in my opinion, only be understood in a systems biological way. So as I mentioned, I have recently published a book, The Gut-Immune Connection, which deals and gets much deeper depth of the information I've presented to you in this short talk. You can either order the book or you can go to my website, sign up for our Mind-Gut Connection newsletter, or get a lot of information that we provide on this website on these various topics. So thank you very much again for allowing me to give a presentation at your conference. And I hope this will have been beneficial and interesting to some of you. Thank you.